Hey everyone, I wanted to give you, those of you that may have missed class or want to do a little review on carbohydrates and lipids, a chance to just look at today's slides. So today we started out looking at two monomers of carbohydrates called monosaccharides, and we talked about some similarities and differences. So these are both carbohydrates. We know they're both made of carbon, hydrogen, and oxygen. They're both in a ring shape, and they are both monosaccharides. However, they both have the same structural formula, C6H12O6. However, they are different in that the one on the left has a hexagon shape, the one on the right has a pentagon shape, and because of their differences in shape, they have different functions as well. So the one on the left is glucose. It is the main energy currency for all living things. And the one on the right tends to be a bit sweeter. It's fruit sugar or fructose. So carbohydrates, the basic rundown is they're good for, all the monomers, all the monosaccharides have the same formula. It's for every carbon, there's a water. And so that's why they're called carbohydrates, right? They're hydrates of carbon. They're a carbon and a water. And they are good for quick energy. So they're what we use for energy, all living things as their energy currency. And then for polysaccharides, when you link a bunch of them together, they give molecules, many organisms, their shape. So the most common polysaccharide on Earth is cellulose, and it's because all plants use the polysaccharide of starch to give them their shape, to make them rigid and able to stand up to wind and rain and all of the other things. For animals and other organisms, they can also serve as signals or receptor sites for responses. So we talked about the monomers of sugars are called monosaccharides. Here are some examples. And you can actually link two monomers together, two monosaccharides together to form a disaccharide. And some of the most common ones are sucrose, which is table sugar, lactose, which is milk sugar, and then maltose, which is a kind of like a malty kind of sugar. And then we can link many monosaccharides together to form a polysaccharide. And there's three that you should know, okay? So here are the three structures of them. Remember, each of these little hexagons represents a glucose. So glucose, 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 all linked together through what kind of reaction? A dehydration synthesis reaction, you got it. And so these will form our polysaccharides. Now, which one do you think would be best to build something really strong and sturdy like a house? If you said this one, I agree with you. And this represents cellulose. So it's packed really tightly. It's in these long lines. And they're actually linking, there are bonds that cross-link the glucoses together as well in cellulose, which is great for plants because, again, it gives it that amazing structure, but it also makes it a challenge to break down. And most animals lack the enzymes to break down cellulose, which is why it's sometimes called insoluble fiber or like indigestible fiber. It's important for us. It helps kind of scour our intestines as it moves through. It helps keep you regular as far as your digestion goes. But we're not able to really digest it. So animals have evolved different adaptations to deal with this, like cows and deer and ruminants have multiple stomachs, and they just like bring it up and chew it again and bring it up and chew it again, right? And that's called rumination. Like if you really think on something, if you're really considering something, people say, oh, they're ruminating. So they're really re-chewing it and rethinking it. Rabbits have a part of their intestinal tract called a cecum, and that is where some of the cellulose actually gets packaged and excreted as waste, and then the rabbit eats that part again. So it's kind of like a rumination, but except it actually like it's fully excreted before they eat it again. Termites have a protozoa in their gut, a single-celled organism that lives inside their intestines and can break down cellulose, and so the termites can harvest the energy from the protozoan, that's the single-celled organism's reactions. And so yeah, but for the rest of us, we don't spend a lot of time eating grass because there's no point, right? We're not getting anything out of it. When plants store energy, like when they have more glucose than they can use, they store it as starch. And you can see, unlike cellulose, starch has a branched structure. Mammals, humans, other animals that store their extra glucose, we don't store it as starch, we store it as glycogen, and that's even more branched than starch. So 3 polysaccharides you need to know in brief: starch, energy storage for plants; glycogen, energy storage for mammals. We store it in our liver and our muscles, and cellulose is good for our diet and a good structural material. All right, we did that. Okay, briefly, lipids are not as clear cut, like here's the monomers, here's the polymers, here's what you need to know. They are really any macromolecule that has a hydrophobic part to it or a nonpolar part to it. That's why we classify the lipids together, is at least some part of them is hydrophobic or nonpolar. So there's a lot, as you can imagine, if you're just talking about like, okay, so what do nonpolar molecules do? Well, they do a lot, and they do a lot of different things. So for one thing, they are long-term energy storage. When we have more glycogen than we can deal with, than we can store, we'll convert those carbohydrates, those polysaccharides, those glucoses in our diet to fat, to triglycerides, okay? They also act as insulation. So, you think of like whale blubber or something like this. It helps against us getting really cold. The phospholipid bilayer here, the lipid part is in yellow, is the outer covering of every cell in our bodies, of every cell that exists. So they're really important. It's really important to have healthy fats in your diet because they make up the outer covering of every one of our cells. They also serve as chemical messengers or hormones. So a certain class of hormones, not all hormones, but a class of hormones are called steroids or sterol hormones. And so in that word, you see the same roots, right? Like estrogen, testosterone. So you see that like ester kind of, that's the same part of the word cholesterol, which is the lipid class that is these chemical messengers or hormones. So vitamin D, serotonin, all of these act as sort of chemical messengers in the body, and they have a lipid basis. Lipids can also serve for cushioning, which is slightly different than insulation. I think of insulation as like protection from cold, and cushioning more as shock absorption. So if you ever feel on the bottoms of your feet, you can find these little fat pads that help with absorbing shock. I mean, just think about before shoes, how hard walking on earth would be. And then finally, lubricants or waxes that are on the surface of every leaf and in things like joint fluids, synovial fluid, etc. So lots of functions. I made a little concept map here that these slides are shared with you that you're welcome to use if it helps you. It sort of helps me break down, 3 main classes of lipids. We'll look at triglycerides first, then phospholipids, and then we'll wrap up with cholesterols. So triglyceride, as you can see in the name, it has three of something, tri, and those three things are three fatty acids. Okay, so you can see here the fatty part, the non-polar or hydrophobic part, is this long carbon-hydrogen chain. And they can be a lot longer than this too. They just cut it here to help see it. So just like oils and that we use to power our machines, those are just pure carbon hydrogen bonds, and we call those hydrocarbons if that's all that's in it, and those store a lot of energy. So for, just think of like the machines, the energy that we use to power our cars and electricity and every natural gas, like all of these things are using hydrocarbons. And that's a big part of this. So these store about 9 calories per gram for humans, which is more than proteins or carbohydrates at about 4 calories per gram. They're a very good energy store. They do have a little carboxyl group, this carbon double bonded to an oxygen with a hydroxyl group on the end. And that's where the acid part comes in. So it does have a slightly acidic quality here. And that's going to bond to a glycerol through what kind of reaction? We're linking two monomers together. I'm hoping you're saying dehydration synthesis. The other clue here is that this is the part that's going to drop out, right? The water. If you're dehydrated, you lose water. So the H2O here is going to fall out. The carbon will make a new bond with this oxygen over here. And then we'll have our triglyceride, a glycerol and 3 fatty acids connected together. Okay, so within triglycerides, there's 2 varieties. There's saturated and unsaturated. And you've probably heard of saturated fats and unsaturated fats before. So what does it mean? What are we talking about when we say saturated? We're talking about the number of hydrogens. So for example, this is a fatty acid, and it has all the hydrogens that could possibly be there are bonded. So every carbon has a single bond to another carbon. Remember, carbon always wants to form 4 bonds. So every carbon also has 2 hydrogens bonded to it, making it completely full of hydrogens or saturated with hydrogens. And so that's why we call fatty acids, we call these saturated fatty acids, because they're full of hydrogens. Now, this will tend to be solid at room temperature because of its straight structure. The fatty acids are all really straight because they're saturated with hydrogens, and they're all single bonds. And so these tend to be the fats that would be solid at room temperature, like the white parts of meat or butter or like any kind of animal fat is usually solid at room temperature. If we take a look at unsaturated fatty acids, what do you notice that's different here? You'll see a double bond between a carbon or more than one carbon. And because of that double bond, you can't have more hydrogens than what's shown here, because carbon, again, it only forms 4 bonds. So with the double bond, that's already 2, 3, 4. We can't have any hydrogens down here. And as a result, the shape of the molecule kinks or bends, which makes it take up more space, which makes it less dense at room temperature. So these tend to be liquid at room temperature as a result, and they tend to be things like oils, plant fats, right? Olive oil, the part at the top of the peanut butter, you know, that's the liquid part. Sesame oil. Coconut oil is an interesting one if you're thinking of that. It actually does have a little bit more saturated fat in it than others, so that one can be solid at room temperature. But for the most part, if it helps you, I put an asterisk just to say, like, this isn't always the case, but tend to be more liquid at room temperature. All right, so try this one. Which do you think is which here? Okay, the one on the bottom is the unsaturated. The little double bonds are highlighted in red. I'm sorry, yellow. So that's going to make these kinks in the molecule, which you can see here. You can't fit as many of them into one space, and so they are not solid at room temperature. Okay, just one more note before we move on. You may have heard of trans fats. So trans fats are unsaturated fats that have been forced into a straight shape. So they're unsaturated, they have the double bonds, but they've been chemically forced into a straight shape, which means that they can pack really tightly, which makes them solid at room temperature, but it also just makes them really unnatural. And so they're really, we don't know exactly how unhealthy they are, but they've been banned in several states. And you just have to be careful when you look at packages, because sometimes they can say 0 grams of trans fats, but they still contain them. It's just that they've kind of played around with the serving size to make it like, well, it's like less than 0.5 gram, so we can call it 0 if we get a big enough serving size. So on the label, what you want to look for is partially hydrogenated. So why does that make sense? Well, it doesn't have all the hydrogens. It's only partially hydrogenated. But it's not, it's a trans fat if they've forced it into this straight shape. So another name for those is partially hydrogenated oils or fats. So check for those. I think Maryland is working on legislation to ban them if they haven't yet already. And then one other thing just of interest, you know, what are omega-3, what are omega-6 fatty acids? We hear a lot about those. That's where the double bond is. So if the double bond happens on the 3rd carbon, it's called an omega-3 unsaturated fat. And those are really important, we think, research shows, for diet. So we should all be trying to increase our omega-3s. Omega-6s are, obviously it's at the sixth carbon. And they are more common and less, we think, less beneficial for diet. Okay, let's move on and talk about phospholipids. As you can see here, they make up the cell membrane. They come in they orient themselves into a double layer, and they look kind of similar to triglycerides, right? They still have a head, and they have they have fatty acid tails, but here's a difference. There's only 2 tails. In the triglyceride, there were 3, right? And in the triglyceride, the head was a glycerol molecule, and in the phospholipid, it's a phosphate group, or a phosphate group head. So the fatty acid tails are still nonpolar. They're hydrophobic. They don't wish to interact with water, but the head is hydrophilic. The phosphates have a negative charge, which makes them more polar, more oriented with water. So in an organism that's 60 or 70% water, like ourselves, the hydrophobic parts, the tails, are going to face away from water, right? It's all water out here outside the cell. It's all water in here inside the cell. So the hydrophobic parts, these fatty acid tails, are like, no water, I don't want to be anywhere near you. And so they always kind of orient in towards one another, and the heads face out. Here's a little cartoon that I thought was cute. The tails do not like water. They wish to avoid it, and the head is charged, so it likes water. We'll talk much more about the phospholipid bilayer in Unit 2. All right, finally, steroids. We gotta have them. We need some chemical messages up in here. What do you notice about their structure? They all have 4 carbon rings. Remember, if they don't say it's a carbon, you just assume that at each of these little vertices there is a carbon here. And then from that, then you can figure out how many hydrogens there are, right? So here's a carbon here. There's 3 bonds to other carbons. This one must have 1 hydrogen coming off of it that's not shown. Here's a carbon here. It has 1, 2, 3, 4 bonds already, so there's no hydrogens coming off here. All right, so they are important chemical messengers. They are what's called hormones, which are just chemical signals or messengers. But these ones are nonpolar, which means they don't interact with water. And so they're things like, but they help produce vitamin D, which is also coordinated by sunlight. They help with secondary sex characteristics, like with estrogen or estradiol and testosterone. And so these are our lipid hormones, our steroids. Okay, that's all for today. Please let me know if you have any questions. Next time we'll move on to nucleic acids and proteins. Thanks, everybody.